I am Dr. Ila Shukla, and this presentation would cover about the preservation of pharmaceutical products using antimicrobial agents. So, the content that I included in this presentation are the pharmaceutical preservatives, why we need to preserve these pharmaceutical preparations, what are the ideal preservatives that we should use, what are the performance requirement of a particular preservative, what is the preservative capacity, how much it can preserve, the factors that will affect the preservative efficacy or the availability, the classification of the preservatives, the antimicrobial preservatives, what kind of antimicrobial preservatives are being used, the preparation required antimicrobial what kind of preparations require antimicrobial preservatives, what are the factors that affect the efficacy of the antimicrobial preservatives, the examples for them, the criteria and the different dosage forms. So, the first slide says about what are the pharmaceutical preservatives. Preservatives are the chemical substances used to improve or amplify the shelf life of drugs by increasing or lowering the oxidative active or excipients by reducing the microbial production. Preservatives are the substances which are added to a particular preparation when we need to check the microbial contamination. This microbial contamination could be due to the particular active substance that is being used, like it could be like if we talk about the ibuprofen, if it is a painkiller, the ibuprofen is the main molecule, maybe the particular microorganism requires to survive upon that particular active drug. So, we need to preserve it. So, we can add because of it, or if other excipients are being added and those are prone to the microbial contamination like talc, kaolin or some kind of such substances are often prone to the contamination by fungi. So, that is why we add preservative just to avoid any kind of microbial contamination, or just to check, just to keep a check over those microbial contamination, that it should not go beyond the limit that it is no more fit for human consumption. Coming to the next slide, it says about why we need preservatives, what is the need of preservation. We have prepared a pharmaceutical preparation, we have done it with the complete awareness, we have done it with a complete caution, but why even then we are adding the preservative? We need to add the preservative, there are the reasons: to protect our drug from microbial attack, to stabilize a product. Often it happens that the certain kind of chemicals that are being used in the pharmaceutical preparation are not actually meant to be kept together. For that purpose we need preservative, so that such kind of interaction may not happen. Then comes to why we do is to enhance the activity and the efficacy of the drug. Moreover, preservatives may also help the drug to produce a particular action. Maybe it could be like an antagonistic effect or a synergistic effect. To increase the shelf life of our product, to increase the shelf life, that means to store our product for a longer duration. What happens when the pharmaceutical preparation is done? When the preparation comes to our use, we use it like if we talk about a multi-dose container, it could be several tablets within a particular container or syrup. When we start consuming it, what happens that we need to uncap it every time we consume it and we cap it back. So, the microbial load that is present in the environment comes in direct contact with that particular substance or a syrup or the tablet. So, for that purpose we need to add preservative, so that the preservative keep a check of microbial contamination, or it does not allow those microbes to grow into it. Then comes another point which says that to enhance the activity and the efficacy of the drug. More often it happens the particular drug is not able to perform a particular action in that given excipient. Maybe the excipient which is being used is not good for that, is not particularly used for that particular drug, but there is no other option for us to use some kind of other excipient. So, for that purpose we add preservative and then we add excipient, so that the interaction is not there and the efficacy is enhanced. So, coming to the next slide. First is ideal preservative. What are ideal preservatives? Like there are particular characteristics which are seen to decide for a particular kind of preservative which should be used for a pharmaceutical preparation. So, first is the effectiveness, how much effective it is there, like how much microbial contamination it can stop or it can check. Second, compatible with the drug components. It should not be there that the preservative added to a particular preparation start reacting with that particular medicament. If it happens, the pharmaceutical preparation will be rendered inefficient, or it should not be showing that kind of potency which is it required to show. Then comes odorless. It should not be having any kind of odor. If it is having a particular odor, maybe it may not be suitable for that particular pharmaceutical preparation, because many kind of patients are there and some patients could be like they are allergic to the smells. Then it should be tasteless. Also it should be tasteless. Why? Because the particular kind of medicament may be having its own taste. Or if we talk about the pediatrics, then the pediatric patients are very inefficient, or they are not compliant for having particular kind of pharmaceutical preparations just because of taste. So it should be tasteless, because it should not add to the bad taste of a particular medicament. Then it should be cost effective. When we go into the production of a large scale pharmaceutical preparation, it has to be cost effective. Then highly soluble. It should be highly soluble, because if it is not properly soluble in case of syrups, or in case of clear solutions or linked acids or something like that, then in case of that the granules may appear, and the granules should not be there in a particular preparation, which need to be clear. Like if we talk about the eye drops, the preservatives used in the eye drop has to be highly soluble. It should not be producing any granule, because if slightest of granules are there, it can damage the eye. Then it should be non-toxic and non-reactive. Obviously, it should be non-toxic. If it is toxic, it is not fit for our consumption, and non-reactive, that means it should not produce any reaction upon consumption. Then stable. Preservative has to be stable, until only then it can be used for a particular pharmaceutical preparation, because preservatives stabilize the pharmaceutical preparation. If they themselves get unstable, then the pharmaceutical preparation will be rendered unstable and unfit for our use. So these are the characteristics which an ideal preservative has. Like first is effectiveness. It should be effective, compatible with the drug component, it should not react with that drug component. Then odorless, there should be no smell. Tasteless, it should not possess any taste. It should be cost effective. It should be highly soluble. It should not be producing any toxic effect. It has to be stable, and it should be having proper effectiveness. Coming to the next slide. So it also says about the ideal preservative. Ideal preservative must decrease the percentage of microbes and prevent any regrowth. It is a must for a ideal preservative or any preservative that it should decrease the microbial growth. The microbial growth in presence of the in presence of the preservative has to be decreased. If it is not decreased, that means the particular preservative is not fit for use. Moreover, the regrowth of the microbes should not be there in the presence of preservatives. So, for that purpose, on that basis, preservatives could be of two types, maybe static and maybe cidal in nature. Static means that they just stop the growth of the microbes, and if the preservative is present over there in the preparation, the microbes are unable to grow. And the cidal effect is the one when the preservative is added into the preparation, they directly kill the microbes in them and they do not allow their further growth. Then some preservatives are ineffective with some microbe strains and should be combined with other to be effective. What happens? There are different type of preservatives, there are different properties of the preservatives. If the particular kind of preservative is not that suitable for a particular microbe, it should not be used, because it would not be able to stop the growth of microbes in that efficient way. Like benzalkonium chloride, like organomercurial, cetrimide, chlorhexidine, and many more examples are there. Coming to the next slide, it says about the performance requirement. What are the performance requirement for a particular preservative? Antimicrobial activity, it has to be active against microbes at low concentration. See, when we go for a pharmaceutical preparation, when the point preservation comes, and the dose, the tablet, when we like take the example of a particular tablet, we add some kind of preservative in it to stop the microbial contamination. What happens is that we do not, if we do not notice that the 2 mg of the dose with 10 mg of the excipient is being used, there is a small tablet and the 1 gram of the preservative is needed, that would be like unsuitable for use. It is totally not suitable, because that size of the dose, the size of the capsule would increase, that would not be easy for a person to consume. So, it has to be keep in mind that the concentration of the preservative that is required for preservation should be low. Then comes aqueous solubility, should be soluble to reach the minimum inhibitory concentration. What happens if the preservative is not soluble enough? Like I just told in the previous slide that it has to be highly soluble. It has to be highly soluble. Why? If it is not soluble, then in the particular preparations it would not be fit for use, and it would not be able to reach the minimum inhibitory concentration that is required for the pharmaceutical preparations. Then come partitioning behavior, remain in the continuous phase in multi-phase products. When we are using multi-phase products refers to like emulsions. If we are using emulsions, we are adding preservative. It should not be a case when we use emulsion. We need to shake it well. If the emulsion breaks out, or if there is any changes in the emulsion, it should not be like that. The preservative forms a different layer. If it is forming a different layer, the preparation is not being preserved. Then organoleptic properties, order and acceptable taste during the administration of the product. As I just told you that it should not be having any order or taste, but it is never like that we are unable to meet all the ideal characteristic, but the order and the taste should be that much in the range that these are acceptable. Then what is the preservative capacity? Preservative capacity is a term used to describe the cumulative level of contamination that a preserved formulation can tolerate before becoming so depleted as to be ineffective. प्रिजर्वेटिव कैपेसिटी की जब हम बात करते हैं तो प्रिजर्वेटिव कैपेसिटी का मतलब होता है कि वो क्यूमुलेटिव लेवल या वो कंबाइंड लेवल आपके प्रिजर्वेशन का या कंटामिनेशन का जिसको कि एक प्रिजर्वेटिव कैन कीप वेल इन विद इन रेंज, जबकि आपका पूरा का पूरा प्रिजर्वेटिव डिप्लीट हो जाए. इट्स लाइक द कंप्लीट कॉन्सेंट्रेशन ऑफ द प्रिजर्वेटिव इज डिप्लीटेड, बट बिफोर दैट इट इज एबल टू क्लियर आउट द माइक्रोबियल पॉपुलेशन फ्रॉम देयर टू अ लेवल दैट इट रेंडर्स इट फिट फॉर यूज. So, next slide says about the factors that affect the availability of the preservative. It's like we have added the preservative is present in there, but that does not mean whether it is showing the preservation or not. So, how much level of preservation is being shown, and what are the factors that would be affecting its availability are active ingredient. What are the active ingredient of a particular pharmaceutical preparation? What are the excipients that are being used or the additives that are being used for the formulation, and the other components. So, classification of preservatives is based on either on the action, like these could be antioxidants हो सकते हैं, antimicrobial agents हो सकते हैं और chelating agents हो सकते हैं, या source के आधार पे naturally obtained preservatives हो सकते हैं या फिर synthetically prepared preservatives हो सकते हैं. Then comes your antimicrobial preservatives. It is added in product to minimize the risk of spoilage and to kill low levels of contaminants introduced during the storage or repeated use of the multi-dose container. I am again and again saying that multi-dose container just because multi-dose container expose that particular preparation directly to the environmental air, and environmental air is having several microbes. So, there are 100% chances of contamination, but here comes the role of preservative. What preservative do? Even if that preparation comes into the direct contact of the environmental air, it will kill the microbes, or it will keep a check that the microbes will not be able to grow in that formulation and it will be fit for our consumption. So, these when we talk about antimicrobial preservatives, these are added into the product जिससे कि जो risk है हमारा contamination का that should be minimized to a level which renders it fit for use. These agents mainly work by inhibiting the cell wall. Now, these are the methods or these are the procedures how these preservative work. Either they can inhibit the cell wall formation नहीं हो रहा है, cell membrane growth नहीं हो रहा है. Cell wall formation is the wall, cell wall is the outermost layer, cell membrane is the layer that is present just beneath the cell wall. Cell wall formation, if it is not there, the defense system of the particular microbe get hampered. Then cell membrane growth is not there, or the other bacterial organelles which may attack our product. What happens when the preservative is antimicrobial? Either it will not allow the cell wall to form, maybe the cell membrane growth inhibited. Cell membrane growth is inhibited, the organelle synthesis would be inhibited, the particular kind of organelles required by these bacteria may be inhibited. Then coming to the next slide, preparations required antimicrobial preservatives. Why it is so, why we require these preservatives has been clear. Now, which kind of preparation are in a requirement of the preservative are solutions, suspensions, emulsions, topical preparations like cream or gels, injectables and eye drops. Why we kept it like solution, suspension, emulsion? Because all these products are having liquid medium, these are having all those substances that are very good for the microbial growth. These are the substances which are source of energy for different microbes, and that is why different microbes used to would love to grow in solutions. If they get a chance to enter the solution and the preservative is not there, the solution would be definitely there would be spoilage, complete microbial spoilage would be there. So, we need a preservative to preserve such solutions, so that the microbial growth is either stopped over there, it is having a static effect, or it is having a sidal effect, that it is not allowing them to sustain over there. Then why injectables? Because injectables are being directly injected into our body, they are either going into the blood stream or into our muscles or at the subcutaneous level. So, for that purpose, if preservation is a very important thing to be kept in mind, because a slightest of microbial contamination may cause a very dangerous effect in it, because all the defense systems are being crossed while we are using injectables. There is no defense system, like if we ingest a substance, the 1.3 pH of this acid is there in the stomach, but it is not the case in case of the injectables. So, preservation is a very important factor that has to be kept in mind. Then eye drops, eye is also very sensitive, so we need to preserve it just to avoid any kind of microbial contamination. So, coming to the next slide, it says about the factor affecting efficacy of the antimicrobial preservatives. What are the factors that affect the antimicrobial efficacy of these preservatives? First is chemical structure. The particular kind of chemical structure of this antimicrobial preservative may not go with that particular kind of active drug or the excipients. The dose, what are the amount of active ingredient or the excipients are there, or what is the dose, like it may require 1 gram of preservative to do the complete preservation and we add just 0.5 mg. So, it is not that sufficient. Then physicochemical properties of the drug, maybe it is in solution form, it is in suspension form, it is in cream or gel. Then type and the level of microbial contamination. If the microbial contamination, it could be different microbes like gram positive, gram negative, fungi, mold, viruses, algae. What are the type of microbial contamination? And level. Level means what is the range, like if it is slight microbial contamination or if it is high microbial contamination. Then the design of pack. Design of pack refers to if it is a strip of tablets having tablets into it, or it is a multi-dose container, like a different tablets placed in a particular bottle or something. Then storage temperature for finished product. What is the storage temperature, if it is, whether it is being capped at that particular temperature or not. Then the range of its function, whether the particular range, whether the particular time is sufficient, or like shelf life is there, like if it is for 1 year, the preservative has to function accordingly. If it is like that 6 months, within 6 months the preservative would disintegrate, then the preservation would not be within that range. Then coming to the next slide, these are the examples of antimicrobial preservatives. First is benzoic acid. It is a food preservative, it is used for preserving several foods. It inhibits the growth of microbes by including mold, yeast and some bacteria. It can inhibit the growth of several microbes including mold, yeast and some kind of bacteria, and it is also antiseptic. It reduces the cases of sepsis. Then coming to the next slide, these are the examples of antimicrobial preservatives. Second one is the methyl parabens. Methyl parabens are white crystalline product. Crystalline refers to the small crystals are there. It is just like you see the sugar. So, sugar is having a squarish crystals. So, such kind of crystals are not squarish, it could be of any shape, it could be needle shaped or anything. So, crystalline powder it is, and it is characteristic order freely soluble in water and alcohol. See, it is just as we talked about the ideal preservative, it is having all those characteristics. Slightest of order is there, it is not having, almost nil order is there, very slight, which is not non-irritant to the person, which is not able to use any kind of odor. Like there are many persons who are allergic to particular kind of odors. So, this kind of preservative would be good because it is having very slight odor. Freely soluble in water, if it is freely soluble in water, it can go with several preparations because it is easily soluble, no crystalline or the crystal powder would be there. And it is also soluble in alcohol. Several type of substances use alcohol for the preparation. So, if it is freely soluble in water and alcohol both, then its range of use of as preservative increases. It is used as antiseptic and preservative in various pharmaceutical preparations. It itself is being used as antiseptic and it is also used in different kind of pharmaceutical preparations. Then it is also used in cosmetic preparations susceptible to decomposition. There are several cosmetic products which have such kind of natural ingredients or something like today you can read different pamphlets like having turmeric and everything like that. So, when we talk about the natural ingredient, they tend to decompose in a particular time. If we talk about the chemical substances, we know the time that they will decompose. The natural substances are more prone because their structure is natural, they are coming directly from the nature, that their kind of interaction with the other chemicals is different. So, they decompose more fast, like they are having more carbon and hydrogen and oxygen molecules, and they are good source for such kind of microbes to survive. So, for that purpose, to avoid such kind of decomposition in them, we add these kind of methyl parabens. Most of the cosmetic products, most of the creams that are being used by you are having these kind of paraben preservatives. Then coming to the next example, that is of antimicrobial preservatives, that is of sodium benzoate. This is the third example. These are the major antimicrobial preservatives that are being used over a range of pharmaceutical preparations. That's why I am discussing here. The sodium benzoate is a white crystalline solid. It was a powder. Methyl paraben was a powder. It is a solid soluble in water and alcohol. This is also soluble in water and alcohol, used extensively as food and pharmaceutical preservative. You must have heard about benzoic acid as well as parabens. You go and you see the cosmetic products, it is paraben over there while we see for the preservative. And when it comes to sodium benzoate, if you consume ketchup, you just find out what kind of preservatives are added in it. Sodium benzoate is there. So, there are several food items and pharmaceutical preparations which are using these kind of the sodium benzoate as preservative. So, it is not a bactericidal one, it is only bacteriostatic agent along with a fungistatic activity. Why its use is range is broad? Because it is fungistatic as well as bacteriostatic. That means it will stop the growth of bacteria as well as fungi, but its effect is not cidal. Coming to the next example, these are the alcohols. Chlorobutanol, it is used as preservative and bacteriostatic agent. It is used as preservative. Why? Because it is having bacteriostatic agent, and chlorobutanol is a very common preservative. It is used in several kind of liquid formulations or solutions. Then you comes the mercurial compounds. This is phenyl mercuric nitrate. It is used as bacterial preservative in pharmaceutical preparations and also uses topical antiseptic. When we talk about topical antiseptic, the phenyl mercuric nitrate is present in it as a preservative, and it also find out its preservation efficiency in many kind of pharmaceutical products just as bacterial preservative. Then comes, what are the criteria for antimicrobial effectiveness? For the category 1, if we say, like bacteria, the 3 log reduction in the 14 days and no increase in up to 28 days, that the preservative should be showing 3 log reduction within 14 days, and up to 28 days there should be no growth beyond it. Then comes yeast and mold, no increase in initial count at 14 to 28 days. Within the range of 14 to 28 days there should be no increase in their growth. Then comes the category 2, the bacteria, the 2 log reduction in 14 days, there should be a reduction of 2 log cycle or 2 log phases within 14 days, and there should be no increase up to 28 days. Then you come yeast and mold, there should be no increase from initial count at 14 and 28 days. There should be no new microbe developing at 14 up to 28 days. Coming to the next slide, the criteria for the antimicrobial effectiveness for the category 3. The bacteria, 1 log reduction in 14 days, with 1 log reduction, 1 log phase reduction within 14 days, and there should be no increase up to 28 days, like it is kind of the effectiveness test for the antimicrobial preservative. Then comes the yeast and mold, there should be no increase in the initial count. What was the initial count at 14 days and the 28 days? We check at 14 days and we check at 28 days, there should be no increase in that initial count. For the category 4, there should be no increase from the initial count at 14 and 28 days. And for the yeast and mold, no increase from the initial count at 48 and the 28 days. What are these 48 and the 28 days? We check on these days. We check at 14 days, we check at 28 days. Then coming to the next slide, the preservatives for different dosage forms. For different dosage forms, there are different types of preservatives that are being used, because the kind of flora, the kind of microbes inside our body at different places are different. So, we use preservative according to that. Like for the rectal one, we use benzyl alcohol, benzoic acid, sodium benzoate, methyl hydroxybenzoate, chlorhexidine, gluconate. For the nasal one, we use benzalkonium chloride, phenyl carbinol and chlorobutanol. These are the preservatives which are used in nasal preparations like nasal drops and all. Then the dental ones, the sodium benzoate, benzoic acid, methyl and ethyl parabens. Then ophthalmic ones, benzoic acid, chlorhexidine, sodium perborate and the boric acid. You must go through that pamphlet which is placed on the eye drops. You will find out such kind of preservatives in such preparations. Then this was all, and thank you.